The title, and really the main finding of our research, is that pre-training neural networks on molecular simulations is helpful for modeling the mapping between protein sequence and protein function. Protein language models have revolutionized how we can represent proteins and make many types of predictions. Most protein language models are based on evolutionary data. In contrast, our method, called METAL, is based on molecular simulations. The motivation for this work is to do protein engineering, where we want to modify proteins to have a desired function. Scientists have created labeled datasets that contain tens, hundreds, or thousands of protein variants, and each variant has an associated functional score, telling us how functional that variant is for a specific functional property. Our goal is to create a model that can predict the functional score given the protein sequence. Then we can use that model to explore the space of possible sequences and find the ones with high function. The challenge is that many labeled datasets are small or biased, which limits the ability of a machine learning model to generalize based on that data. We want to overcome these challenges by utilizing molecular simulations. Like I said before, many other methods use evolutionary data. That data contains a strong signal, but it's not necessarily ideal for protein engineering, where we might be interested in variants that wouldn't appear in nature. Our method is called mutational effect transfer learning, or METAL for short. Step 1 of our method is to select 150 diverse base proteins from Protein Data Bank. Then we generate 200,000 variants for each protein, where each variant has 1 to 5 amino acid substitutions. This gives us a sample of the local sequence space around each of the 150 diverse base proteins. Then we run those variants through the Rosetta molecular simulation software in order to compute energy terms for each variant. The energy terms capture different aspects of protein stability. Step 2 of the method is to train a transformer model to predict the Rosetta energy terms. This model learns an internal representation of protein sequences that's based on the Rosetta energies and the input data. Step 3 of the method is to transfer the learned representation to experimental data and fine-tune the model to predict the specific functional score from the experimental data. We also tested a protein-specific version of METAL trained on variants of a single protein of interest. We refer to this protein-specific version of METAL as METAL local, and the version trained on diverse protein variants as METAL global. So far, we have evaluated our method on the GB1 deep mutational scanning experimental dataset. Looking at small training set sizes, the two versions of METAL outperform the baseline linear regression and sequence convolutional models. METAL also outperforms ISM1B, a state-of-the-art protein language model which we used as a feature extractor. Looking at the extrapolation results, METAL local outperforms the other methods we tested across 3 different types of extrapolations. With position extrapolation, METAL local performs substantially better than the other methods, perhaps due to its GB1 specific pre-training data. With mutation extrapolation, ISM1B is the second best performer, followed closely by METAL global. With regime extrapolation, the CNN is the second best performer, again followed closely by METAL global. In conclusion, The main takeaway is that pre-training on molecular simulations improves protein sequence function modeling. Looking forward, we are continuing to develop this method and train more advanced and better performing source models. We plan to compare METAL to more variant function prediction methods, as well as evaluate across a wide range of sequence function datasets. Thank you for listening, and please feel free to reach out with any questions.